We'll begin this discussion with a little bit of a review of how DNA sequence is generated. We've already worked quite a bit with DNA sequence from GenBank and looked at alignments and talked about how we might analyze that for phylogenetics, but let's talk about where that sequence comes from. Now, originally, the very first DNA sequences were generated via something called Sanger sequencing. Sanger sequencing is very similar to a PCR reaction, except there is a small number of defective nucleotides, the building blocks for DNA, that are put into the reaction. And so when one of those defective nucleotides is incorporated into the chain of DNA that's being built, it terminates the sequence. So sometimes this is called termination or di-termination sequencing also, but Sanger sequencing is a little bit more common. And so when that occurs, we end up with a piece of DNA of very specific length. And that is done over and over and over again, and so we get many, many sequences that terminate at the same place. Now, originally when this was first done, they had to do 4 separate reactions, one for each of the different base pairs, and then they would run them out on a gel and manually read them off. And so notice the shortest one here that's run the fastest through the gel is a G. It's in the G column. The next one is an A. There is a C, there's a T, another G, and so on, and so they could read the sequence that way. And that was very, very labor intensive, both for the chemistry, setting up the reaction, and also for actually physically reading it and having someone sit down and do the sequence. But it was a breakthrough. And this was in the late 20th century. I think around the 80s is when this method was first developed. And then very rapidly it was replaced with a similar but more advanced development here. So it's still Sanger sequencing, but they were able to put a different label on each of the 4 nucleotides. So they could run a single reaction, and then computers were developed and technology machines that could read the individual absorption spectrum of those 4 different sequences, and so we could just go down and we could read it like this, and even algorithms, computer programs were written that could do this automatically, and so generate the DNA sequence without having to have a person there. And so although the chemistry was still fairly prohibitive, not quite as bad as before because it was 1/4 of the amount, but it's still a lot of work. Largely we automated the sequence reading itself. And so Sanger sequencing was great, and that was again in the late 20th century. In the 90s going forward into the 2000s, we began to have this technology available. And so we were able to generate way, way, way more sequence than had ever been generated before. And so this really was the onset of genome sequencing. And initially small genomes were sequenced, and eventually larger ones that we were interested in. We tend to be a little self-interested, so the human genome was one of the earlier ones. A lot of information in the human genome. About 3.2 billion base pair is coded there in those molecules. And so the human genome sequencing project took about a decade, give or take, hundreds of millions of dollars, and the first human genome in draft form, just kind of raw sequence form, was published in 2001, so about 20 years ago. Now, more recently, within the last decade, we've had a dramatic breakthrough again in how we sequence DNA. I'm not going to go into the details or the chemistry of this. I'm not going to ask you to memorize them. I think you should probably know them in general outline form. That's why I've provided this link for you in the PowerPoint. You can look at it. It's a little 5-minute video that explains the most common type of what is called next-gen or high-throughput sequencing. There are other forms, some of which were developed and are kind of being abandoned. Others are still being worked on. But the idea is that we can sequence massive amounts of DNA, and this is often called a shotgun approach. And the early genome sequencing with Sanger sequencing were also a shotgun approach, and I'll review that very, very brief outline of shotgun sequencing in the next lecture. So don't worry about it if you don't know what it is. I'm hoping you do, but I'm keeping this somewhat basic because I realize a lot of even graduate students don't have a clear understanding of this. And so if you do have questions, email me or jump on to one of the Zoom sessions. You can also look them up, and there's a lot of good information, but sometimes it's tricky to sort through all of the vast amounts of information that are freely available on the internet. Everything you ever wanted to learn is freely available now on the internet. It's a remarkable age we live in. And so you're like, well, why am I taking this class, Dr. Terry, and what good are you? Hopefully, I am able to serve as a moderator, a synthesizer, and give you information from all the vast amounts that's most relevant, and maybe do it in a order and in a way that's going to make sense to you. So anyway, you can look it up, but if not, that's fine. Look at this video for next-gen sequencing. Now, the end result of all of these methods, at least with a reference to genome sequencing, is a vast, vast amount of DNA data. And we have to figure out what to do with it. But this vast amount of DNA data is little snippets, little bits and pieces from the entire genome. And so the very first thing we have to do after we do sequencing is we have to figure out how to assemble it. So this diagram, the blue line represents the entire genome. The red lines represent the pieces that we've sequenced. Now, the way that next-gen sequencing works, we actually get paired reads that are on the end of longer sequences of DNA. So that's why we have the little red boxes with the blue lines there. So the red boxes represent sequences that we know. And notice how some of them overlap. There are parts where we have sequenced the same thing more than once. And so we can line those up and find, if there's enough of an overlap, find the matching sequences. And eventually we can assemble the entire genome. Now, to do this, because it's randomly, these little snippets of DNA are generated randomly. Some of them we sample many times, some portions. Other portions we may sample only a few times. And to ensure that we have very, very high coverage, up into the 99th percentile, we need to do about 50x sequencing. So for instance, the human genome is about 3.2 billion base pair in its haploid form. And to be able to sequence that, we would need to randomly sequence the equivalent of about 150, 50 times 3.2 is, right? 156, so basically 156 gigabases, or 156 billion base pairs of DNA, we would need to randomly sequence so that we would have about 99 or more % coverage of the entire human genome. And then we would need to take all those random little bits that we have sequenced and assemble them in this manner. Okay? So, this can be done. There are algorithms that do it. DNA sequencing is getting faster and cheaper. When this first started, it may have cost you tens of thousands of dollars and months of time to sequence the entire human genome, even using state-of-the-art next-gen sequencing. But today you could sequence an entire human genome for a few $100s and assemble it with relative ease in just a few days. Okay, so you could have that sequence and assembly, and in fact people are doing this. And so we're generating vast, vast amounts of DNA and really swamping out, by comparison, everything that was generated for decades during the first generation of DNA sequencing. Now, this does not mean that assembly is trivial. It still is a fairly labor-intensive, and we'll look a little bit of that probably in an upcoming project. But it is still a very labor and computationally intensive process. But we have programs, we have algorithms that do make it much, much easier, especially compared to what was done previously. Now, one thing that can also make it more easy is if we have a reference assembly. So for instance, if we are looking at multiple human genomes, we could take the first human genome that we've assembled and use it kind of as a ruler, because it's going to be largely identical to any other human genome that we sequence. And there are going to be differences, allelic variations, a single nucleotide polymorphisms, any other mutations that might be represented in that genome that we're looking at. And that's the reason why we sequence so many genomes. We want to get an idea of the variety and the diversity and hopefully correlate that with interesting diseases, interesting phenotypes, or any other thing that we might be able to look at. Now, one last thing that can make this assembly difficult are repetitive sequences. It turns out that many genomes have large chunks of DNA that are repeated over and over and over again. These can be as short as 2 base pairs in a row, maybe repeated a dozen or several dozen times. Or they can be much longer regions of DNA that many of these were created by transposable elements, or transposons, that are represented many, many times in the genome. And so that makes assembly difficult, because if you have these little short reads and many parts of the genome that are identical, those short reads could line up to any one of those identical portions of the genome. And so that makes it challenging, and for those portions of the genome, which usually don't contain protein coding, at least ones that are going to impact the organism's phenotype. So they're not as important in that way, but they still are important to get the entire genome. So we might need to do much longer reads and use different methodologies to try and figure out those repetitive portions of the sequence. So after all this is done, we end up with a portion of the genome. So, of course, with the humans, for instance, there are 23 pairs of chromosomes. They range in size from tens of millions of base pairs to in the hundreds of millions of base pairs. And then all together, we put them together, and we have 3.2 billion base pairs representing that haploid genome. I've got a little bit more than 30,000 base pairs here on this slide, and I know it's going to make your eyes hurt to kind of just scan at and look at it, but I just wanted to kind of get it here for impact factor. Realize this is a tiny, tiny fraction of the human genome. And so assembling it is only the first step, and it's a little labor intensive, but once we've done it, then we can move on and we can begin to say, okay, we've got the raw sequence. What is here? What can we find here? And to do that, we need to understand the genetic language. We need to be able, once we've assembled it, to be able to do what we call mapping and annotation. So let's just give those definitions, and there is a little bit of overlap, but in this class we'll use the term mapping as simply identifying features on a genome. It might be a gene, might be a repetitive element, it might be a centromere or a telomere, but we just simply identify and maybe highlight or make a note of where those features are located. And then annotation refers to adding additional information about that feature. It might be a gene, there might be several allelic variants of it. It might have different splicing end results of the gene might be spliced together in different ways. So any one of a number of additional information is just simply saying, oh, here is a gene. Okay. Now I would also like to talk about how we identify genes, and this is something where there is a lot of misinformation. Even advanced biology students often have very little idea about how we would find a gene. And realize this is not a trivial task, especially initially when we had almost no idea about where these genes were found. Now luckily, before we had entire genomes, people had been doing a ton of work on human genes. They'd been targeting them specifically using polymerase chain reaction and different methods to look at just that one gene. So we knew many of, maybe even most of the human genes before we had the human genome. And so we could take those and look for their matches on the human genome. But even that was not a trivial event. And so one of the most common things when people say, okay, here I've got a lot of raw DNA sequence. How do I find genes? Almost always the first answer I'll get from biology students is say, oh, you just look for a start codon. All genes start with a start codon. That's ATG at the DNA version. Okay, yes, but there's a little bit of a catch-22 or paradox here. So let me just do this. I'm going to find ATG in this sequence, 40,000 base pairs. This, by the way, only represents one gene in here. But I'm going to look for where we have ATGs. And so I say find next. We'll say, well, it's going to highlight them. There's 1, 2, 3, 4, 5, 6, 7, 8. In fact, as I do this, you'll notice there are dozens and dozens of ATGs across this sequence, but I told you it's only one gene. Why do we have so many ATGs present? Well, recognize that in non-coding sequence, ATG is going to pop up 1 out of every 64 possible 3 base pairs in a row is going to be ATG. And so we're going to just have lots of them, and an ATG can be a methionine in the middle of a gene. An ATG can be a non-coding DNA and not be a start codon at all. An ATG can be in the middle of a gene, but maybe the A, for instance, is at the end of one codon, and the T and the G are the beginning of the other codon. And if we're out of reading frame, we would look at it and say, oh, ATG start codon, but it's not a start codon. It's two components of different codons. Okay, so we cannot look for start codons for the same reason we cannot look for stop codons. So it doesn't allow us to find genes in raw DNA sequence. So how do we do this? Well, it's a little technical and complicated, but there are 3 really good, easy ways. Of course, once we've identified them, if we're just sequencing another genome like a human genome, we already know where to expect to find the genes. All humans have the same genes, barring some sort of major mutation, right? This is the important difference between a gene and an allele. We all have the same genes, barring any major mutations. However, we have different alleles for those genes, which what makes us genetically different from one another. So if I know where the gene is that controls eye color, for instance, I can go to that same part of a different person's chromosome, different person's genome, find that exact same gene. I might then look at, oh, is it different in what maybe allelic variants there are? But I know exactly the address, the locus of where to find that gene if I've already done it in a previous genome. Okay? But if we have an unknown genome, it's a little bit more difficult. Even if we have a genome of closely related organisms, it's not going to be the same, maybe even not very much close to the same, and so we would need to look at different methods. Now, when I say the same, you always hear these things about, oh, the human genome is 90 some odd % similar to the chimpanzee genome. That's true at the DNA level, but when we look at where those changes are and how they have impacted, there can be some major differences, and we can have major rearrangements. We'll look at that in a coming lecture here. Okay, so I'm going to add to this PowerPoint, I'm going to add 3 methods, and there are others, and it gets a little computationally complicated. But here are 3 easy ones that we can say that would help and have provided a huge input into finding genes. Okay, so let's put a little bit of new text box in here. So number 1 is we can look for some feature that is unique enough to a gene and long enough that it's just not going to pop up randomly. And so although start codons and stop codons don't meet those criteria, we do have some things that do. And so the best one is, say, we could look for promoters. Promoters at the beginning of each eukaryotic gene, they're long enough that they don't often pop up just by random chance, and they're conserved enough so we can have a single sequence to look for across a wide range of organisms. Okay, so we could look for a promoter. That would help us. Number 2, and this was the most common one that was done early on, we could look for open reading frame. And that's abbreviated ORF. So write it down in your notes or make a note in your head, however you can best remember it. ORF stands for open reading frame. And an open reading frame is a long stretch of DNA where in one of the 6 possible reading frames there are no stop codons over a long stretch. So just by chance, about 1 out of every 16 or 17 base pairs is going to be a stop codon. And so if we see a long stretch where one of the reading frames doesn't have a stop codon, that's a really good sign that there's a gene there. Okay. Now, the final one that we're going to do here, and this is only available more recently, and we'll talk about these at a later date, but we could map a transcriptome onto a genome. The one downside of mapping transcriptomes onto genome is that in a single transcriptome, we don't have every single gene represented. So we might need multiple transcriptomes. But the cool thing is that that is all of the coding region. Now, coding region is only 1.5% of the human genome. It's a very small fraction of all eukaryotic genomes. But it's a very critical and important part, one that many people are interested in because it has a massive impact on the phenotype. Okay, so let's put all those there so you can see them. Just again, review them, make sure you understand that. We can't just look for start and stop codons, but using these methodologies, we can begin to identify genes. Once they're identified, people can do more work on them and annotate them and understand them, look for allelic population diversity, and those things are ongoing. Now, because of also the massive amounts of DNA, we are beginning to have comparative data sets where we can look at a human genome and a chimpanzee genome and an orangutan genome and a gorilla genome and maybe even many other primate genomes. And as more and more become available, we understand more and more of the trends and the forces that are shaping the evolution of genomes.